In this video lecture, I will be talking on precipitation, interaction between antibody and soluble antigens leads to lattice formation, which developed into a visible precipitate, as can be seen here. This is an interaction between soluble antigen and soluble antibodies. So soluble antigens combine with soluble antibodies to form insoluble visible complex. Antibodies lead to formation of visible precipitate are called precipitin. Lattice formation depends on antibody valency, which is number of antibody binding sites. And as can be seen here, this is IgM. It is formed of 5 antibodies connected together by the joining chain, and these are the binding sites which allow binding to 10 small molecules, or here it is known as decavalent, or it allows binding to 5 big molecules and act as pentavalent. In case of IgG, it has 2 binding site, so it is bivalent or bivalent. Accordingly, the antibody must be at least bivalent, as in case of IgG. Similarly, antigens must have 2 copies of the similar epitopes, as can be seen here. These are monoclonal antibodies, so the epitopes are at least 2 of them are similar, as they allow 2 monoclonal antibodies to bind. However, in case of antigen with different epitopes, cross-link lattice structure with polyclonal antisera are formed when the antigen contains multiple different epitopes. So soluble antigen and soluble antibody make insoluble product, as can be seen here. This is the line of precipitation. So this is useful, or it is used for diagnosis of infection by viruses, bacteria, protozoa, or detection of toxins, toxoid glycoproteins. And here, this is very clear, the precipitation curve, as the precipitation takes place at the site of equilibrium or zone of equivalence. So the number of antibodies here are equal to number of antigens, and this will lead to lattice formation. However, in zone of antibody excess, the reaction is negative, and in zone of antigen excess, the reaction is also negative. So precipitation also can be done in a solid agar matrix, and it is used to detect and measure relative concentrations of antibodies or antigens, and to compare antigens, or to determine the relative purity of an antigen preparation by 2 methods: Mancini method, which is radial immunodiffusion, and Ouchterlony method, which is double diffusion precipitation method. And here, this is the radial method. The antigen is placed in an agar in this well, So they diffuse towards the antibodies at the periphery, and they form a line of precipitate, as can be seen here in blue, when the antibodies and the antigens they bind together and at the zone of equivalence. In case of Ouchterlony method or double diffusion method, the antigen are placed here in this well and antibodies placed in another well, so they diffuse together, and at the zone of equivalence they form a line of precipitate. Thank you very much, Professor Ahmed Molla.